Good afternoon, everyone. Today I'll present our results on exosome signaling in colorectal cancer. Exosomes are a specific class of nanoparticles secreted by a wide range of cells. They are typically 40 to 100 nanometers in size and are synthesized by a complex and enzyme-dependent pathway. They are understood to play a role in cancer progression by the transfer of cancer-associated nucleic acids and proteins. In colorectal tumors, there are cancer cells surrounded by the supporting stromal tissue. The heavily stained cells are colorectal cancer cells, and stromal tissue is mainly made up of fibroblasts. It is well established that cancer cells can secrete signals that influence fibroblasts, and that likewise, fibroblasts can also influence cancer cells. Exosomes are a novel class of nanoparticles that are important in this two-way transfer of information. Our first objective was to isolate exosomes from both colorectal cancer cells as well as fibroblasts and demonstrate their transfer to recipient cells. We then investigated the cellular effects of colorectal cancer exosomes on fibroblasts, as well as the cellular effects of fibroblast exosomes on cancer cells. When colorectal cancer cells and fibroblasts are grown in culture, they release exosomes into the culture media. To isolate exosomes, culture media was harvested, put through serial centrifugation, and gave us an exosome pellet. When visualized under transmission electron microscopy, we can see that our exosome samples are between 40 and 100 nanometers in diameter and have intact lipid bilayer membranes. Our exosome samples were also positive for known protein markers such as TSG101 and CD63. Colorectal cancer exosomes can be transferred to fibroblasts. Here are some fibroblasts under normal microscopy, and after the transfer of colorectal cancer exosomes that have been labeled with a green lipophilic fluorescent dye. Exosome transfer is a two-way process, and exosomes can also be transferred from fibroblasts to cancer cells. Here are some colorectal cancer exosomes that have been engineered to express a red fluorescent protein, and from their cross-sectional views, we are able to see if any particles have entered these cells. After the transfer of green fluorescent fibroblast exosomes, we can clearly see that they have entered the cells, and this view is even more apparent in the cross-sectional images. We know that exosomes can be transferred from fibroblasts to cancer cells and vice versa. We wanted to see if they play any role in the key signaling pathways of cancer. Many of us are familiar with the hallmarks of cancer, and one of the key reasons for treatment failure is when cells resist cell death mechanisms. The AKT pathway is a key signaling pathway for cell survival. In normal circumstances, AKT is phosphorylated and active, and BAD is phosphorylated. Therefore, cells survive. In dying cells, AKT activation is lost, and BAD is not phosphorylated, and cells die. Chemoresistance is directly linked with how much BAD protein is phosphorylated by AKT. In chemoresistant cells, there is an augmentation of BAD phosphorylation. Now, these proteins aren't named for no reason. Too much phospho-BAD will be bad for you. Another key process that's dysregulated in many cancers is proliferation signaling. The RAS-ERK pathway is an important signaling pathway for cell proliferation. Under normal circumstances, many extracellular growth factors converge onto the RAS protein and activate ERK signaling. In many cancers, genes on this pathway are mutated, such as RAS, leading to an overphosphorylation of ERK and increased cell proliferation. Western blot shows us that cancer exosome transfer influences fibroblast protein profile. Firstly, colorectal cancer exosomes do not induce ERK activation and therefore cell proliferation in fibroblasts. However, colorectal cancer exosomes induce AKT activation, and more specifically, they induce the AKT-BAD phosphorylation pathway. What this means is that stromal fibroblasts may become more resistant to apoptosis due to the acquisition of colorectal cancer exosomes. When exosomes were transferred the other way around, from fibroblasts to cancer cells, this also influenced the protein profile of recipient cells. Firstly, fibroblast exosomes induce AKT activation, and specifically they induce the AKT bad phosphorylation pathway. This correlates to a protection from chemotherapeutic agents. In the second and fourth columns, we see that colorectal cancer cells have a reduced cell death after treatment with oxaliplatin if they were co-cultured with exosomes from fibroblasts. Secondly, fibroblast exosomes also induce ERK activation in colorectal cancer cells. As the ERK pathway is the key regulating pathway of cell proliferation, we see that this Western blot result corresponds to the enhancement of proliferation in colorectal cells after fibroblast exosome transfer. Our key conclusions are that firstly, colorectal cancer exosomes can be transferred to fibroblasts in vitro and vice versa. The transfer of colorectal cancer exosomes induces AKT bad phosphorylation in fibroblasts, which may promote resistance to apoptosis. The transfer of fibroblast exosomes induces AKT bad phosphorylation in colorectal cancer cells, which has been shown to contribute to chemoresistance mechanisms. Lastly, the transfer of fibroblast exosomes induces ERK activation in colorectal cancer cells, which allows enhanced proliferation. Thank you.